How can we explain the properties of our visual consciousness based on the architecture and mechanisms in the visual system? Our naive visual phenomenology actually makes sense. That's the sort of the conclusion from the last video. But there was some problem. And one of the reasons is that to understand why our introspection about color in the periphery makes sense, we need to understand the mechanism that explains that, right? So far, the evidence of mechanism suggests that we don't have much cones at the periphery. So how can it be the case that we can make an accurate judgment about color similarity in the periphery and fovea or periphery and periphery? So to explain that, we need to understand the important concept of in the neuroscience, which is called the receptive field of the neurons, and also its properties. Okay. And a receptive field of neurons in the periphery is, in a short summary, much bigger than the receptive field for the neurons in the fovea, and that basically pretty much explains this phenomenon. All right, so this is the data that I showed in the last slide from Ariel's study. So this basically shows that central and the peripheral vision color similarities structure is the same. And here, just to recapitulate, the size of the stimulus is quite small. It's the roughly 1/10 of the thumbnails, size. That's the fovea, a 0.1 degree visual angle. So it's much smaller than the demo video has actually shown. And peripheral ones were 4.5 times bigger. So it's still like half the size of this, 1/3 of the size of the thumbnail, and that was presented around 10 degree to the periphery, and still we see this nice structure. What happens if we use a very small stimuli at the fovea and the periphery? And then it indeed degrades quite a bit for the peripheral related structure. So here, within the central comparison, because foveal stimulus is the same size, the left side comparison structure and right side is the same. However, the periphery to central comparison or periphery to periphery comparison became much closer to something really blurry, and many things becomes kind of similar, rated as 0 or 1 or 2 or 3. And it's quite unlike this, sharp structure here, periphery or periphery central. And why is that? Why does the size matter? That's the next question. Right? So to understand this, we need to understand this critical concept of neuroscience, receptive field. So as I said in the last week, retina has a bunch of neurons, and as an output from the new retina, there's a retinal ganglion cells that makes spikes like this. And then experimenter can penetrate the electrode like here, and then to record this spiking activity. That is explained in chapter 3 of the Quest for Consciousness. And just remember that the cones and the rods are, at the back of the retina, and the eye receives the light like this way. So there's a scatter of the light because of the ganglion cells and so on. This is quite strange, but that's how our eyes works. All right. But then the property of the receptive field of retinal ganglion cells. So on center cells, is a one type of the retinal ganglion cells, okay, in the retina, and that fires. By the way, x-axis here is always a time over 2.5 seconds, and when you don't present any stimulus, and then record the activity of the retinal ganglion cells over 2.5 seconds, while stimulus period is here, then these on-center cells tend to fire some kind of background activity here and there, and this vertical line is the spiking of the neurons. But it doesn't do much. And then here, when we present some kind of white spot at the location of the vision that corresponds to its receptive field, then it starts to make a firing like this. On the other hand, if we present the stimulus too big, even though it includes these highly sensitive areas, then it starts to become silent again. And then if you remove this part by black, then it becomes completely silent. And this means that these on-center neurons tend to have an excitable receptive field component here inside, and then inhibitory surround. That's the structure of these on-center cells. On the other hand, something similar but opposite polarity has been also reported. That's the off-center cells. And off-center cells, like on-center cells, as a background, it doesn't fire at all if it's uniformly black. And if it gets presented by the small spot of the light, white spot in the center, then it actually inhibits its response completely. And then there's a rebound here as well, rebound here. And then if it's completely white all the way, then it doesn't fire indistinguishably from the baseline. And if it's presented with a white ring, so it's the opposite of this on-center cells, then it starts to fire like crazy and it becomes off. So this complementary kind of response makes this on-center and off-center cells. Here, at the center, there's an inhibitory region, and the surrounding is the excitatory regions of this on-center cells. Okay. And this has to be explained in the end by the structure of the ganglion cells. So ganglion cells have this extended dendrites at particular location in the retina. If there's if I enlarge my retina like here, then there's some kind of neuron here, and then that extends the dendrites. And the dendrite collects the input from the cones and rods at each location. And then here I'm showing 6 different kinds of the cells. There are 2 types of cells, like midget cells and parasol cells. And the midget cells are the ones that receives pretty much the input from the cones. Okay, and then parasol cells are the ones that receive the input from the rods. Note that both of the cases here, that number 1 millimeter, 5.5 millimeter, 5.8 millimeter, or 10.5 millimeter, that is the distance from the fovea in the eye. Okay, so 10.5 millimeter means that the 1 centimeter off from the fovea. And then with that, the size of this dendrite, extent of the dendrite becomes really big. And this is the size of the neuron. So it's still small, but you can imagine that at the fovea, closer to the fovea, the size of the receptive field becomes very, very small for both cones and the rods. Okay, this is important. So just to recap, size of the retinal ganglion cells and its extent of the dendrite becomes bigger and bigger at the periphery. Okay, and these are the determinants of the extent of the receptive field. How, which areas of the visual field do these retinal ganglion cells respond to? Okay, and the important thing is that the wider dendrite collects inputs from retinal ganglion cells across the wider areas. These cones and rods. What it means is that this is a slice of the retina that I showed last week. So the light comes from top to the bottom. Like this way. And then it gets collected by these rods and so cones and so on here. Okay, photoreceptors. And then these information gets propagated through these, retinal cells called bipolar cells and amacrine cells and so on. Almost like vertically, like here. But eventually, when it comes to retinal ganglion cells, these information gets collected by this extent of the dendrite, right? So if there is one retinal ganglion cells like this or this, big, then it extends the dendrites like here. And so the areas of the visual field that contributes to this, ganglion cells are this extent. And then depending on the inhibition or excitation, for example, this one, if this is the on-center cells, then that one collects the excitatory information around here, and then inhibitory information around surround. That's the on-center cell. But the important thing is that the extent of the dendrite becomes bigger as the retinal ganglion cells locate in the periphery. Okay. And then next important thing is that the dendrites are wide at the periphery, but this is not only limited at the level of the retina. Okay, it's a common property across layers of the visual processing that I'm going to explain later in this lecture video. So if this is the eyes, and then if I'm looking at this particular visual stimulus that goes magnifies across a visual field, then let's imagine what happens using this schema. So all these small stimulus that projects to the all these small stimulus that is around this fovea, okay, that projects to here. And then all these retinal ganglion cells has a very small receptive field, or sort of the defense area, right? It only pay attention to one particular area, very small spot, and therefore these guys actually can respond to this particular red stimulus, and then it gets maybe if it likes red long wavelengths, it shows a very strong activity, and if it's not, then it becomes silent and things like that. And that sends an activity to lateral geniculate nucleus in the thalamus, next relay station, and then that converges to one particular neuron in V1, the primary visual cortex that I'm going to explain next. That means that it's like retinal ganglion cells, which collects lots of neurons, lots of input from the cones and the rods. B1 neurons also collects lots of input from the LGN that corresponds to many different areas of the visual field. And this kind of convergent kind of shape, hourglass-like structure, happens across many layers. That's one point. Okay, that's across layers. Another thing that is important is here. So what happens when you pay attention to this in particular peripheral retina? And this one is looking at, let's say, these particular areas of the yellow, right? And then each of these retinal ganglion cells have a larger receptive field size, and or each of them can collect this light corresponding to yellow, right? And it can pass the yellow information to the next layer, and then B1 can also collect this yellow information as such. And so there has been no degradation of the color information as long as the size of the stimulus becomes bigger for the periphery. And that matches with the property of the receptive field size of the peripheral neurons. Okay. So to summarize about up to here, neurons that respond to the peripheral visual inputs reflect the inputs from cones over wider areas than those that respond to the foveal visual input. Because at the foveal, its receptive field is very small, but at the periphery, it captures lots of cones and cones outputs. The convergence effect gets stronger for the neurons at the higher visual areas. So even at the level of retina, there is already some kind of convergence, and that becomes bigger in the visual cortex, and that becomes even bigger as you go in the higher visual hierarchy. And the visual hierarchy is the next topic in this video. So assuming that the color experience arises from somewhere in the higher areas, and that's a part that I'm going to explain in the next few weeks, then the big and also uniform surface should evoke stronger color experience. So even though I said that maybe there's a color of the blue color in the sky that is memorized or expected, and that guarantees this blueness in the sky, there's no necessity to invoke that kind of expectation or memory, in fact, because blue sky in the periphery, as long as it's big and uniform, then the peripheral retinal ganglion cells can actually collect all these, uniformly blue information and then project it to the cortex, and then you can experience this, nice and vivid color of the blue. Okay, then what happens if the stimuli are small and fixed size at the and also located randomly? And that was one of the demonstration that I showed also last week and also in the beginning of this lecture. So when you see something like this, you probably saw a color relatively clearly only on the fovea, but not in the periphery. And you even right now, you may be able to say that, oh, this is, while you're fixating here, I can see something light here, bright here, but I can't really see the different shades of the color around here if you don't move your eyes from here, right? And why is the case? That's because, again, in the at the fovea, each of these small stimulus can project it to the corresponding retinal ganglion neurons and generate this nice red sensation. On the other hand, if you go to the periphery, then each of the peripheral ganglion cells become kind of stimulated by different types of the wavelengths of the information. And as a result, then even if each of them likes to respond only to the yellow, it doesn't respond as vividly as possible. And that's related to the LGN, and then as I said, B1 further converges this kind of thing. And this convergence even further, happens at the larger areas, later areas. And as a result, this kind of small and fixed size stimulus does not generate vivid color sensation. Okay. So to sum up, if colored stimulus are small, fixed in size and random, then neurons at the fovea have a better spatial resolution, and that gives us still the color sensation at the fovea. But collecting inconsistent color information at the periphery predicts more blurred responses of the neurons responsible for color experiences at the periphery. And so that explains why we can see the color vividly if it's big and constant and uniform, but not if it's small. And taken together, you can say that we cannot see color at the periphery. Okay, as long as the stimulus is the right condition, we do see color at the periphery. And to come back to this, misleading figure, I'll just make one point, right? In general, this figure is not really explained in many cases of psychology or neuroscience, textbook. One of the issue is that the y-axis here is the density, number of the receptors per square meter. And also another thing to notice is that it doesn't go to zero. Zero is here. Okay. So as you can see here, within this so-called area called blind spot, both cone and also rod become 0 because there's only axons. But all the way through, cones are present at the lower density. What it means is that when the density needs to be can be interpreted, it needs to be multiplied by the size of the area, right? To estimate the number of the cones that contribute to the process underlining it. So as I said, as you go to the periphery, throughout all the visual hierarchy, peripheral neurons have a bigger receptive field size. And as a layer of the processing goes up, this kind of convergence effect becomes larger and larger. So this statement that color cannot be experienced in the periphery needs to be revised. If you learned it in the psychology class or neuroscience in the past. Okay, so in sum, retinal ganglion cells send visual inputs via spikes through RSGN to V1. And the size of the receptive field of a retinal ganglion cell scales with the eccentricity. It becomes bigger and bigger at the periphery. And the same is true for all the visual areas. And this can explain why we can see colors of big dots at the periphery, but not small ones. Okay.